Now let's turn to the next session, which will focus on inactivated COVID vaccine. As we know, China is the main country developing multiple inactivated COVID-19 vaccines. Next, let me introduce the chair for the next session, Professor Joon Rhee from Chonan National University Medical School from South Korea. Dr. Rhee is also a fellow and a board member of ISV. So it's my great honor to introduce Dr. Rhee, please.
Thank you. Thank you, Shan, for kind introduction. I'm Joon Rhee. This is native Korean pronunciation. I'm from Jeonnam National University. Welcome to this session 2, which will be dealing with inactivated vaccines. We have 2 speakers. We'll start from the first one, which will deal with COVID-19 vaccine from viral cell inactivated from Sinopharm, China National Biotech Group Company Limited. The speaker is Ms. Meng, who is working in this industry for 16 years, and he has experience in biological industry, including vaccines, diagnostics, and therapeutic products. She led and successfully completed a number of technical partnerships in licensing, NGO funding support, overseas registration, commercial cooperation deals, and including OPV capacity expansion and pre-qualification program with Bill and Melinda Gates Foundation. She'll be briefing their phase 3 trial results to you. Ms. Lee?
Thank you, Chair, for the introduction. This is Lima from CNBG. China National Biotech Group is a state-owned company who dedicated in the manufacture of biologicals. And our major activity is actually research and development, as well as manufacturing and commercialization of multiple biological products, including vaccines, diagnostics, blood products. So today I'll give a brief introduction of our COVID-19 vaccine, and this is a viral cell produced vaccine, and we use very classical approach for the production, which is inactivated. And in this presentation, I'll present data from both Beijing Institute and Wuhan Institute. So please go to the next slide. So my presentation will consist of 3 parts. The first one is a brief introduction of our vaccine and the result of clinical phase 3 study. And I'll also briefly introduce our future plan. Next. Go to next slide, please. So this COVID-19 vaccine contains the inactivated SARS-CoV-2 virus. And we isolated this strain for the production from 3 clinical isolates. And we also use WHO already pre-qualified viral cell line for the production. This vaccine contains 6.5 units per doses of antigen. And the specification is 0.5 milliliter per dose. We have 2 specifications. One is pre-filled syringe, the other one is vial, and we're also working on multi-dose specifications. This vaccine is very user-friendly, which can be stored at 2 to 8 degrees Celsius, and the shelf life tentatively is now 2 years. And this vaccine is formulated with aluminum hydroxide. The target population is 18 years old and above, and we have already completed study in the 3 to 17 years old group, and we are expecting this population will also be included in the target population in the near future. Please go to the next slide. As you all know, in response to the COVID-19 pandemic, we developed this vaccine in a short period of time, but this is based on our existing infrastructure and technology platform for the production of for the large-scale production of COVID-19 of inactivated vaccine. And we obtained the clinical trial approval in China in April last year, and we kicked off the first clinical phase 3 study in UAE in last June. By the end of last year, we obtained the first marketing authorization from UAE and later on in Bahrain and China. Next slide, please. The next slide. Please go next. Okay, so this is the study objectives. The primary endpoint is to study the incidence of COVID-19 starting on day 15 after 2 doses of vaccinations in healthy population aged 18 years and above. And for the secondary endpoint, we are going to study the efficacy against severe and death cases of COVID-19. And also to analyze the GMT and antibody levels. So the adverse events was also collected during the solicited period and the non-solicited period. And for the explanatory efficacy endpoint, we studied the neutralizing antibody and also the ADE and VED. Please go to the next. So this is the overall design of the study. It is a multi-center randomized double-blinded placebo-controlled phase 3 clinical trial. And in this UAE-centered trial, we involved 4 countries in the MENA region. And in this study, we actually included 3 groups. 2 vaccine groups, 1 from Beijing, 1 from Wuhan, and 1 placebo group. The ratio of each group is 1 to 1 to 1. And the schedule we used is the optimal schedule we found during the clinical phase 2, which is 0-21 days schedule. 2 doses regimen was used. So please go to the next. And you can see from this slide that we take blood from each selected time point. And we also completed complete safety assessment. And the follow-up period is 1 year after the second dose. So please go to the next slide. And regarding the clinical classification of the diseases, we use the same criteria of WHO. So next, please. These are the research sites and ASCO approval. So we got from different countries. And we actually included 5 countries into this study, 4 countries in this study: UAE, Bahrain, Jordan, and Egypt. So please go to the next slide. And as of December 2000, by end of December 2010, this trial enrolled nearly 45,000 people. And from this slide, you can see the number of subjects enrolled in each country. So next, please. And these are the demographic data and baseline characteristics from the analysis. And this number shows the number of subjects enrolled in each subgroup, including people with different age, different gender, and nationalities. And the median observation time was 112 days, which meets the criteria set by WHO. Next slide. And this is a second result of the second interim analysis. And this interim analysis was triggered by the number of cases, which is required to be more than 100. And in this second interim analysis, we actually find 142 cases, of which 95 were in the placebo group, 21 were in the Beijing vaccine group, and 26 were from the Wuhan group. Next, please. So this is the primary efficacy results. According to the cases we identified in this trial, the statistician did the calculation, and the vaccine efficacy was approximately 78%. And the vaccine efficacy in the WIVP vaccine group was 72%. Next, please. So this is a subgroup analysis. You can see that for people with different age, different gender and ethnicities, and baseline positive or negative group, the vaccine efficacy is similar, all above 70%, and which shows this vaccine is very effective in all those different subgroups. Next slide, please. So this is the total adverse events incidence. You can see that in the slide, the blue bar, the placebo, and the orange bar is the WIVP group, and the gray one is for Beijing vaccines, which demonstrated these two vaccines. Both vaccines has very good safety profile, and the adverse events incidence is similar among the placebo group and the vaccine groups. The majority of the adverse reactions were grade 1. Please go to the next slide. And you can just quickly go through those slides regarding the safety categorized by time and by different criteria. The most frequently seen adverse events actually pain at the injection site, fatigue, and mild fever. So this vaccine showed very good safety profile similar to other inactivated vaccine. Please go next slide. So this is actually the adverse events after the second dose. Next, please. This is for the 18 to 59 years old group. Also showed very good safety profile. Next slide. Next, please. So for the immunogenicity, if you look at the seroconversion 14 days after the second injection, the vaccine group showed a very high rate of seroconversion in compared with the placebo group. This result works for both neutralizing antibody and binding antibodies. Please next. Here is the GMT result, and you can see that both vaccines can stimulate high level of neutralizing antibody, GMT. So the vaccine groups are significantly better than the placebo group. Next, please. Next. And this is a summary of the second interim analysis. The vaccine efficacy for the Beijing vaccine is approximately 78, and it has a very good capacity to prevent the severe cases of COVID-19 in the age group of 18 years and above. And it also showed very good safety profile in comparison with the placebo. And the adverse events incidence in the population aged 60 years and above is a little bit lower than in the age group of 18 to 59. And the adverse events are mainly grade 1 AE. And for the immunogenicity, it also showed very good seroconversion and also high level of antibodies. Next. And this is a similar slide, shows the results of WIP's vaccine. And the efficacy rate is 72, and similar similar incidence of adverse events was seen in this group as well. Very good safety profile, and it also showed a good seroconversion and high antibody level. Next slide, please. And this is the result for the cross-neutralization. It shows that the serum samples from clinical phase 1, 2, and 3 can neutralize different virus strains from clinical from those prevalent strains. And the next slide. The next slide is from a paper published by Damish and Gaofu, which shows the Beijing vaccine can produce a good capacity to neutralize North African strain. Next one. So this is a summary for the cross-neutralization effect of this cross-neutralization protection results. And the results of the test shows it can neutralize different strains from the domestic and abroad. And it has extensive cross-neutralization reactions to the current epidemic strains. Next one. This is the recent data from the emergency use in China. As of March 31st, we got data AFI reports from 65 million people vaccinated in China. And the common reactions are mainly local reactions like redness, swelling, and for this systematic reactions are mainly fever, fatigue, mild reactions. So it also shows a very good safety profile in mass vaccination campaign. And it also has very low level of abnormal reactions, mainly allergic rash and other mild reactions. So this is the result for the adverse reaction after launch. So next one. So for the future plan, next, we will conduct the pharmacovigilance according to the WHO requirements, collect the data from the real-world use, and do the coding and follow-up and analysis. If there's anything need to be take actions, we will introduce required interventions accordingly. So next one. Next slide, please. So those are the risk management plan. We actually identify those known risks and potential risks from the previous clinical study. And for the missing information, which will be completed through following studies in special populations, and also we will do co-administration studies and other studies as per the requirements of regulatory agencies. We also introduce pharmacovigilance plans accordingly, and mitigation strategy was also established. Next slide. So in China, we are going to conduct active safety monitoring in 100,000 people, and a passive safety monitoring plan is also implemented. It is also ongoing in 1 million people, and we are also going to do the study in the breastfeeding women. and long-term safety monitoring will be conducted. The co-administration and the ADE and VAD will be monitored during the following studies. And of course, we will also complete the study, clinical phase 3 study in UAE and other countries. And the DSMB will continue to monitor the ADE in the field. So next, please. So I'd like to thank our partners in different regions, and also thanks our partners for the GCP study and also the regulatory agencies for supporting us in this study. So thank you for your attention, and if there's any questions, please feel free to let us know. Thank you.
Thank you. Thank you. It's my turn.
Yes.
So do you hear me?
Yes.
Okay. Thank you for your concise presentation. We have a long list of questions. Since you keep the time, we have 10 minutes or so for Q&A. First, I'll start with the question related to previous. Do you have any data about immunity induced after a single dose? How does it compare to 2-dose regimen? Any data about T-cell response?
Okay, thank you for the question. Actually, we do collect the blood samples after the first dose. It is included in our protocol for clinical phase 3. And we also did some preliminary study on that. And according to the data from our clinical phase 3, the efficacy after 1 dose can reach approximately 65%. So that data was shown to WHO stage meeting last time. And so 刚才第二个 question 是什么来着？忘了。
T-cell response.
Oh, okay. And for the T-cell response, actually we did some study during the in the animal study. And we will continue to monitor the results from our clinical trials and to further analysis this aspect. But as you all know, for the inactivated vaccine, it is not supposed to take effect through T-cell response. But anyway, we will do more study on this aspect, and if there's further data, we will share it later on.
Okay, thank you. Well, we have, participants worldwide. These questions should have been from Argentina. The question is, due to limited availability of vaccines, Argentina government decided to extend the interval between first and second dose of Sinopharm's vaccine to 3 months. What does the manufacturer has to say about it? So what do you think about extending the interval between first and second doses up to 3 months?
Actually, for the interval of immunization, it is already started in the clinical phase 1, 2, and also in clinical phase 3. And the schedule we recommend is still 21 days, or 21 days schedule with 7 days window period, which is from 21 to 28 days. And if you give injection with this interval, it can stimulate the best immune response. So that's why we still recommend the government to use the original schedule. And the schedule with 3 months was not started, so I cannot, really recommend you to go this way. And for the supply of vaccines, we have already expanded our capacity for the production. And now for the capacity, we can reach 1 billion doses by end of this year. And later on, I think in the later half of this year, the supply will be more sufficient in comparison with the first half of the year. So we are expecting to supply more doses to both COVAX and other countries.
Okay, thank you. You have tried two strains, one from Beijing, the other from Wuhan. Why did you do that?
Actually, in CNBG, we have 6 institutes for the production of vaccines. So we have more than 100 years of history. And those institutes, they develop those vaccines independently. And this is actually a backup strategy to make sure that during this emergency response, we can have at least one vaccine available for the world and for China. So this is why we developed these 2 vaccines in parallel. Actually, we also have another vaccine candidate which just got approval that is a recombinant vaccine. So you can see that this is our company strategy to mitigate the risk. And we also produce these 2 vaccines in 2 different sites, and we are also doing bulk filling in our other institute just to make sure that we have sufficient supply to the world and also to COVAX.
Okay, main reason was risk mitigation, right?
Yes, risk mitigation.
Yes, and another question. Some people in the UAE are getting a 3rd shot of your vaccine. Is this a 3rd booster shot? Is Sinopharm actively evaluating the efficacy of 3rd booster shot? Do you have any plan or ongoing study?
Actually, in our clinical trial, we do need to continue to monitor the immunity persistency. And we have been prepared for further studies, but the decision has not been made because we are still doing the test of the blood samples from 6 months. So for the UAE 3rd dose issue, it is actually not included in our clinical plan. I think this information is from the emergency use. The country made their own decision that they found for some immunocompromised population or for the elder groups, if they found they have low level of antibodies, they may give them a 3rd dose, but it is not recommended in our formal schedule. And in addition to that, as per the requirements from regulatory agencies, and also you can see from the news, Moderna, Pfizer, they are doing additional dose to evaluate the efficacy against the variants. So I think in the future, we will also need to take that into consideration.
Okay, thank you. Next question is about combining your vaccine with other companies' vaccines. Since
sorry, Chair, I cannot hear you.
It should be inevitable, combining different companies in a vaccine. Do you have any plan for that kind of study, or do you have any experience with combining with other companies' vaccines?
Yes, we do have plans for the future sequential use of different type of vaccines. And we also have planned to conduct trials to study the co-administration with other vaccines like pneumopolysaccharide vaccine and also with influenza.
Yes. Another question is for your efficacy with new mutants. So you didn't present that data. So do you have any data for protection against newly emerging mutants from, for example, South Africa or Brazil?
Okay, and in our current clinical phase 3 study, the efficacy against variants was not included. But we do have some laboratory data, which I showed in the slides, that the serum from our clinical phase 1, 2, and 3 can neutralize multiple strains from the field. And in the future, we will also conduct additional study to continue to monitor the efficacy of our vaccine against variants. But in principle, we believe for the inactivated whole virion vaccine, because it actually preserved all those epitopes on surface of the virus, so it can have broader protection. In principle, it can have a broader protection against different variants. But this needs to be proved in the field, so we will continue to monitor its efficacy against the variants.
So this last question could be difficult for you to answer, but there are questions about, the methodology. How did you inactivate coronavirus vaccine strains?
That's beta prophylactone.
I mean the how you inactivated your viruses.
Actually, this is according to our classical manufacturing process. And that manufacturing process has been used in other inactivated vaccines like enteric virus 71 and Sabin IPV. So it is a proven technology which can produce this inactivated vaccine in large scale. And we also have a complete site of testing method to ensure that this virus is inactivated appropriately, that has no virulence, but still retain the immunogenicity to simulate sufficient level of antibodies. I'm not sure whether I answered your questions.
Okay. Thank you for your enthusiastic answers. We'll stop here. We'll change gear to next presentation from Sinovac. This is about phase 3 clinical data and the real-world vaccination of CoronaVac produced by Sinovac. This topic will be presented by Weining Meng, who is the Senior Director of Overseas Business of Sinovac. He joined Sinovac since 2007. He led the first Chinese Hep A vaccine to WHO pre-qualification, and he is overseeing the overseas business in his company. Mr. Meng, please.
Thank you, Chair. And it's an honor for me and to sharing the data for our CoronaVac, and also since the vaccine already be large used in many countries, so we also by collaboration with our partners, we also would like to share some the usage of the vaccine in the real world data. Next slide, please. So basically, Sinovac is R&D oriented vaccine company, and you can see the history. Well, Sinovac is still a young company, and we have 20 years history. Actually, today is our 20 years ceremony, and you can see the history of Sinovac is also a history of making vaccines, no matter it's generic vaccines or no vaccines. So something I need to mention is in 2003 for SARS, we are Sinovac already involved in the development of SARS vaccines. In 2004, we already completed phase 1 clinical studies for SARS vaccines. Although there is no SARS anymore, but actually the development of SARS vaccine is also the fundamental for current CoronaVac, which is same technology, inactivated whole variant adjuvanted vaccines. In 2009, for the previous pandemic, Sinovac is also play a very important role, and we are the first company globally to obtain the product license for H1N1 vaccines. Next slide, please. And currently, we have two major sites location, one in Beijing and one in Dalian. In Beijing, we focused both inactivated viral vaccine and some bacterial vaccines. In Dalian, we will focus on the live attenuated vaccines. And the CoronaVac is located our new site in Daxing province of Beijing. And we quickly finished construction and start use of this new site since August of last year. Currently, the production capacity is over 2 billion doses per year. Next slide. So, although we are quite young company, but actually we already start our footprint in other countries since more than 17 years ago. And currently, our previous flagship hepatitis A called HELIFE already pre-qualified by WHO, and that vaccine already used in more than 20 countries. And I also because this international network, and for this COVID-19 happened, we quickly collaborate with our partners in other countries and working together and to quickly push the phase 3 clinical trial in several countries. Next slide, please. So regarding this vaccine, the development process, we initiate the development for this COVID vaccine on January, and since we have a lot of experience for inactivated viral vaccines, and also we have experience for previous SARS vaccines. So obviously we adopt the inactivated whole variant with aluminum hydroxide as one technical approach. And actually, in parallel, we are also trying to another technical approach, including the recommended protein vaccine, and also some modified live viral vaccine. But after a couple of months, we found the inactivated vaccine can give a much better result. So we go on for the development for these vaccines. Our preclinical studies were conducted in April, and we are the first company to provide the efficacy data on monkeys through the viral challenge studies, and that data also were published in Science at the beginning of May. And the phase 1, phase 2 was start in April and May, and based on the major data, we got phase 1 and phase 2, and we are quickly running our phase 3 in other countries, including Brazil, Turkey, Indonesia, and Chile. Next slide, please. So this is overall for our clinical study program plan, and actually we use the same design for both phase 1, phase 2, and phase 3. It's randomized double-blind with a 1-to-1 placebo control design, and we also took the sequential design for phase 1, phase 2. And trying to save some time. Basically, our after 7 days evaluation on the first dose, and then we start we move to the second step for the different population. Currently, we already have the data from the population over 3 years old, and our phase 3 is conducted in several countries. mainly focused on the adult population over 18 years old, including Brazil, Turkey, Indonesia, and Chile. And maybe most of you already know the data we got from our phase 3. And currently, we are also already completed the breeding studies to evaluate whether there's some difference comparing the pellet production and commercial lots. And currently, we are still in the developing process for some post-marketing, post-licensure studies, including some large-scale real-world studies by collaborating with others, government and other partners. And we are also trying to evaluate the different vaccination schedules. That is in our plan. Next slide, please. So, well, today I will mainly talk about the phase 3, but I also would like to share in just one slide from our phase 1, phase 2. This is the immunogenicity data, including the different population, 3 years to 17 years old, 18 years to 59 years old, and also 16 years population, and also including the 2 different schedules, 0-14-day schedule and 0-28-day schedule. Also, in our phase 1, phase 2, we evaluate 3 different dosage: lower dose, middle dose, which is also our target dose, and higher dose. We summarize all the data here, and we also list our reference here, including the phase 1, phase 2 one for our population. And actually, based on our studies, we found the target dose with 0-28-day schedule can give a best, much better the immunogenicity data. And the neutralization antibody GMP from the pediatric studies from the children population is better than the adult population. For our phase 3, actually all our phase 3 we use 0-14-day schedules, because at the very beginning we considering 2 schedules: 0-14 days as an emergency use schedule and 0-28 days as a routine schedule. And so first step, we are running 0-14 days. Even we found that the GMT titer of neutralization antibody is lower than 0-28. But under current pandemic situation, to let the people and to generate antibody as soon as possible also is very important. Based on that, we are also running the 0-14 days. But currently, the large use of this vaccine in many countries mainly follows 0-28 day schedules, because we know that 0-28 day can give a better immunogenicity. Next slide, please. So I just put some little summary, but I already mentioned, I think, so just go next slide. Okay, for our phase 3 design, actually we are evaluating. We have a similar protocol in different countries, including Turkey, Brazil, Indonesia, and Chile. We use the same batch of the vaccines and also follow the same schedules. And in Brazil, it's quite special because these studies, we only conducted studies on the healthcare workers and who are fighting with who are frontline fighting with COVID in Brazil. So they are really high risk population. A total 13,000 plan to enroll. Turkey is mixture. Around 10% of subject is the healthcare workers, and for another 90% above is general population. In Indonesia, it's a small-scale trial, and the major target to evaluate lot consistency. In Indonesia, our another objective for these studies is get the authorities required satisfied. And Chile, the trial is a little bit different because the trial is running since the end of November. At that time, just several weeks after the Chile government already approved this vaccine for the emergency use. So we have to stop the progress plan and do an amendment and to change, to open the playbook and give the different schedule. So in Chile currently we are evaluating 0-14-day schedule and 0-28-day schedules. Next slide, please. Here is just the demographic calculations of subject in Chile, and I'm not going to talk too much details. And if you want to more details, you can find our publications in the preprint website. Next slide, please. The primary endpoint for these studies is to evaluate the efficacy of the vaccines and also to evaluate safety 7 days after, and also we designed some secondary endpoint and just to evaluate the efficacy on different serious cases, and also have more information and for the different time. And also we are trying to evaluate the risk for disease enhancement. Next slide, please. So in order to evaluate efficacy, the key element is to make the definition for COVID-19 cases. This protocol actually was drafted in June of last year, and we are taking reference from US FDA and the WHO. And so the definition is any symptomatic cases lasting for more, any symptoms lasting for more than 2 days plus PCR positive. We confirm this is a COVID cases. But during the studies, our authorities, NIPA, also give the recommendation on the COVID definitions, a little bit straight than previous one, and they require 2 typical symptoms, or 1 typical symptom lasting more than 2 days, and plus PCR test, and then we'll confirm these COVID-19 cases. So when the data available, we also follow the NIPA guideline because we have to register the vaccine in China. Next slide, please. Another interesting or another significant element we designed for these studies, we are also use WHO clinical progress scale definition in these studies, because the studies are running in the healthcare workers. So no matter the subject or the investigators are very familiar about the COVID cases, about the symptoms, so it's also likely for them to make the more sensitive defined during the clinical studies. So we give a different score to the different the symptoms, including from number 2, from score 2 to score 10, such as the mild cases is 2 and 3, and moderate cases 4 and 5, and the severe cases from 6 to 10. By this way, we can have more information about the efficacy to different grade of the cases for COVID. Next slide, please. So here is the final result, and the general speaking, if including all the systematic cases, the overall efficacy rate is 50.65%. There is some I think after that we do analysis and we found some element for this not high efficacy rate general. One is we found 85% of the COVID cases we collected. Total we collected more than 250 from 10,000 subjects. but 85% and above is very mild cases, which is only score 2. That means 1 or 2 symptoms lasting 2 days without any medical care and plus PCR test. And for the more serious cases, also mild cases but need medical care, we found totally about 40 to 50 cases. And the efficacy rate for that case above others moderate and serious is 83.7%. And totally we found about 10 cases, including hospitalization cases and other serious cases, and 1 death cases. They are all in the placebo arm. So the efficacy based on that date for the moderate cases and above is 100%. Next slide, please. And since we have a lot of cases through these studies, the study is running from July, but the major enrollment actually starts since the beginning of October, and so we really catch the peak season. Most of the cases are reported at the end of October until the December. So at that period, Brazil suffer a very high peak. And we also do some sensitive analysis. Since this is only conducted in healthcare workers, so we did not enrolled many elderly people, a total about 600 above. And so the efficacy from elderly people, only we collect 6 cases, and so the meaning is not that, the meaningful is not high. And another interesting we found, so we follow the 0-14 day schedule, and but actually there is about more than 1,500 subjects. They are the second dose they take on 21 even later date. And based on that, we found actually the longer interval between the phase 1, phase 2 does give a better efficacy result. So about 10% above. And that is why currently, when the large use of vaccine by different countries, we recommend them to follow the 0-20 day schedules. Next slide, please. And so our major data coming from Brazil, and also we already have data from Turkey and from Indonesia. From Turkey, actually, we don't have detailed information. We just have roughly data. I think our partner in Turkey is still clean the database and drafting the CSR. So later we will have more information from Turkey. But regarding the efficacy, actually from Turkey we got a very much higher efficacy data because you can see totally we found 41 cases. But among them, 6 of them are hospitalized cases. So the percentage of very mild cases are much lower comparing to Brazil. That could be one reason why the efficacy is higher. Another reason may be because the strain different, because the dominance of strain in Brazil is different with the Turkey ones. And so the 6 hospitalized cases are all in the placebo arm. So the efficacy for the moderate cases and severe cases, 100% based on that. Next slide. And we are, although the only I think only 1,600 subjects enrolled in the Indonesia trial, but finally also partner in Indonesia collect 25 cases. All these 25 are very mild cases, only 1 or 2 symptoms, without any medical care. So the efficacy rate based on that is 65%. Next slide. And the objective of the Indonesia trial, another major objective is to evaluate lot consistency. So we deliver 3 lots to them, and from the several data, no matter several conversion rate of GMT data, we see the consistency between 3 lots is very nice. Next slide. And next, about 2 or 3 slides is about the safety from these 3 phase 3. I'm not going to tell the details, but actually we did not find some risk from our phase 3, and the safety profile of this vaccine is quite nice. It's similar with other inactivated viral vaccines, and we don't find a significant difference between the vaccine group and placebo group. The major interactions from Brazil trial is the pain on the injection site. Follow that is fatigue and fever and other things. That is also similar with others inactivated with aluminum hydroxide adjuvanted vaccines. Next slide. And from Turkey, similar. The conclusion is similar. So there is no significance between the vaccination group and placebo group. The only difference is about pain on the injection site. In Turkey studies, not many subjects report this as ICE. Next slide. Indonesia safety data also similar with the Brazil data. Also, it's very similar with our phase 1, phase 2. No much significant difference between vaccine and placebo. Painful on the injection site is a major one. Next slide. And one more thing I need to comment that is from no matter Brazil, Indonesia, and Turkey, we did not observe the disease enhancement from the current data. We will go on to safety monitoring and to also evaluate these rates in the futures. So this we put a little summary here, and so we overall efficacy follow the 0-14 day schedules in different countries. We get different numbers about the efficacy, and but most important, I need to emphasize is the efficacy on the mild cases in medical care in Brazil, 83.7%, hospital and the severe cases 100%. And later, I'm also going to sharing some real world data. Also, is also gave the similar result on that. Next slide, please. And so based on those data, and also plus our phase 1, phase 2, and the vaccine Coronavac has been approved for emergency use or conditional marketing authorization by 40 authorities, including China. And up to April 26th, more than 280 million doses has been distributed globally, and more than 180 million doses has been used in the world by more than 30 countries. Next slide. So because this large used in different countries, we have, well, not we, or even the countries has opportunity to conduct some large scale studies. This is the preliminary effectiveness of Coronavac in Chile. And up to now, I think around 90% of the vaccine used in Chile is our Coronavac. Total more than 12 million doses already used in this country, including about 7 million people received the first dose and 5 million received the second dose. And the strategy for Chile government and to use the vaccine from the elderly population. So, for example, from the population over 100 and over 70 and over 60, 50 by this order. And during in parallel, the scientists also monitoring the data because they have very comprehensive healthcare system to monitor, can collect all the cases, COVID-19 cases. So totally about 10 million individuals over 16 years, including these studies. About 3 to 4 million people with the vaccination, about another about 5 or 6 million, I can't remember, without vaccination. And they comparing the outcome between these two groups. Finally got these serious numbers. So 14 days after second dose, they found the effectiveness is about 67%, and but most important for the hospitalized cases is 85%, and for the ICU hospitalized cases is 89%, and for the deaths is 80%. The variation for deaths will be bigger than others because that they only conduct these studies for 2 months. So both us and the investigator for studies think they will need more time and to get more accurate results from the death cases. And I also put the data for the ICU hospital cases data, so we can see after the vaccination, it can reduce the ICU cases significant. Next slide. And another very preliminary effectiveness of our vaccine is conducted in Manaus, in Brazil. This city is quite famous for the mutant P.1, and currently it's more than 75% of COVID sample are P.1. And the scientists also conduct negative case control studies, and so they also first vaccinate about 53,000 healthcare workers. And after that, they collect the cases and to conduct analysis. And this is a very preliminary because the study is still going on, and the data was also published on the Medicics preprint website. So the conclusion is a vaccination with at least one dose can give around 49.6% for the other systematic cases, and also they collect the infection, the effectiveness on the infection is 35%. We will keep monitoring that studies, and also including our Brazil partners. And currently there are also several studies running, some conducted by our partners, some also conducted by different investigators. So I think maybe we need more time, 1 month or 2 months. There will be more data available from different countries. Next. I think that is all the last one. The last one is from healthcare data from the São Paulo city in Brazil. And we can see Brazil follow the same vaccination schedule, same vaccination strategy with Chile. They start the vaccination on elderly people, and this is also the severe cases from their healthcare system in this city. So we can see especially for the elderly over 90 years and 85 to 89, because for that population, most of them already got the vaccinate, and obviously the ICU patient cases were much lower comparing the others population without full vaccination of this vaccine. So that is all I would like to share today, and happy to take any questions. Thank you.
Okay. Thank you for your detailed presentation showing data generated in different countries. I'll start with this question. You have shown differences in the efficacy among different countries. How do you interpret the results?
Actually, we are not trying to put every result and to get some more analysis on that. Because although the protocol is similar, but we have different investigators and studies conducting a different subject. This is like because during that period also the variance or domination is different, and also the different area the risk for the target also different. For example, I mentioned that in Brazil this is a really high risk population, and from the placebo we found the rate report rate 23%, and just several months, and also Brazil has a much higher reporting rate comparing Turkey and Indonesia. So is reasonable, and we got the different efficacy, and considering the strain dominated in different countries also different.
Okay, so partly that could be attributed to the different culture or system in different countries and different prevalence, right?
Yeah, another thing I forgot to mention is if you look at the details, you will see that if you look at the percentage of the cases, in Brazil, we got too many, not too many, we get many very mild cases. And because they are healthcare workers, they are more sensitive. And for example, actually, we define some centers for PCR test, and totally we have 16 sites in Brazil. But when we collect the data, we found all the PCR positive result coming from 87 different hospitals. And so that is also meaning because they are professionals, so they have more sensitive. They also have a more chance to get the PCR test, and by that way, obviously, the percentage of very mild cases will be higher, and they will be also lower than the total efficacy rate.
Okay, same question to you. Do you have any plan or data of combining with other companies' vaccines?
Actually, we don't have this plan, but to be honest, we have some plan, but it's still discussion with the different investigators. For example, China CDC but generally speaking, currently we already found, for example, in Brazil, people already conduct the studies by using our vaccines together with AstraZeneca vaccines. So we are also looking forward to see the more data coming out. But from our side, we are still in a preliminary discussion with the different stakeholders, mainly in China. Over.
Yes, as I mentioned before, because of the global shortage of vaccines, we shouldn't have any other choice than combining different vaccines. So I hope your company and the other companies will cooperate each other in providing more vaccines to developing countries.
Yeah, that's definitely our target.
Yes, and another question is, did you have any problems with failure to seroconversion for seroconversion even after the second dose?
From our studies, we found yes, there because I remember from our phase 2, the seroconversion rate is 95 or 96%. So we do find about 3 or 4%. There's no sero positive, and during the real world, the real use vaccination, also some people told us after the full dose, second dose, and they are going to test the antibodies by some testing diagnostic kit, and they found still negative. But this is some cases we heard that, but we still need more information, because the diagnostic kit by using the ELISA we cannot judge comparing the neutralization antibody.
Okay, another question is, do you expect even better efficacy with longer interval between doses, like 3 months?
Yeah,
21 or 28 days should be better than 14-day interval.
Yes.
So how about extending it to up to 3 months?
Yeah, currently we are considering make some other studies for that. And we are also, by collaboration with our partners, for example, the Chinese CDC, and to collect the real world data. And I think we already have some data, and our partners already submit for the publications. Maybe after 2 or 3 weeks, maybe the data, the detailed data will be published. And from our current information, a longer interval, 3 months, it should be similar, even better neutralization immunogenicity we got comparing the 0 to 28 days.
That should be a good news for many countries.
Yeah.
My last question is, I don't think you show the neutralization data against new variants. So do you have neutralization data against the South African, Brazilian mutant strains?
Yeah, we do have the data. And also this study is conducted by us, also collaborating with other scientists. And the data was also already submitted to the international journal, and I think they will be published later. And so, generally speaking, for the Brazil strain B.1, for South Africa strain B.1.351, we do see the decrease regarding the neutralization antibody titer. About decrease about 70 to 80%. But interesting is we did not find this comparing the P.1 and B.1.351. The result is very similar. And we also actually our Brazil partner Butantan, they also conduct the similar result. They also conduct a similar test. They comparing. They take the blood sample from the phase 3 studies, and I can't remember if phase 3 or the real vaccination. And they do the neutralization on the previously strain from Brazil. It's B.1.28, I remember, and also P.1 and also P.2. And also, they didn't find much different. So the similar antibody titers, and plus what we found a similar result comparing B.1 and B.1.351, and plus we have some, not we, but some scientists in Brazil already conduct the result in Manaus. And they have 75% above is a B.1, and seem to now up to now the result is good. Only major is only 1 dose. Also, some subjects have 2 doses. The efficacy rate already nearly 50%, And also considering the result I present from from Chile. In Chile, the major dominate strain is about 20 to 25% is a P.1, it's Brazil one, but the efficacy rate is still quite okay, and much better even in hospitalized cases. So from total together, our thinking is the efficacy, the vaccine, because this is the inactivated whole vaccines. So that advantage of this vaccine has a broad against to the mutant. So it seems to now we have quite a nice result, preliminary result to against P.1, and also should be a similar result to the B.1351. And another concern about the UK strain, but the UK strain, if you look at the sequence, it's quite similar with our CZ02. So we don't worry about that strain. Over.
Okay, so that should be the advantage of the inactivated whole virus vaccine.
Yeah, indeed.
Take home message is your vaccine showed somehow decreased neutralizing activity against newly emerging strains, but still effective in preventing disease. Okay.
Yeah.
So thank you for your detailed explanation and answers. I'll stop here. I'll close the session 2 and transfer microphone to Shan. Thank you.
Thanks.
Thank you, Professor Rhee, and also all the speakers. I really want to thank Professor Dolan and Professor Rhee to organize today's session, in particular for the excellent presentation by 3 speakers. May I have the summary slides? I want to just use these slides to show where we are in terms of our Congress, also the progress of global COVID-19 vaccine. About 2 months ago, we organized the 4th ISV Congress. We invited 4 companies in Europe, North America, and Russia. The 4 companies received their local government emergency use authorization. Today, we are fortunate to have the full vaccine from 3 companies received similar authorization from China and from other global countries. As we learned today, this inactive vaccine has provided great supply to many developing countries. As we know, there are several other companies in India, in North America, Europe. They have also received a similar authorization. It is ISV's intention to organize continue this series. So we hope in the future our audience can come back to learn more, because we strongly believe at ISV, even though we are living in a very challenging time, it is very important to have a fair, transparent, and scientific platform to exchange the progress of COVID-19 vaccine. Next slide. Because you can see we are still not out of the woods. We can see clearly the 3 countries we list here, United States, UK, and Russia. They had peaks of infection, but because each of this region had a very early aggressive vaccine campaign, you can see the trend, the pandemic is under somewhat control or not rebounding very powerfully. On the other hand, you can see the situation in Europe is quite different. There is a huge hesitancy from the public, from the vaccine supplies, so you can see they are still up and down. Of course, we are also seeing in other parts of the world, such as India, Brazil, due to the resource limitation and also public health control issues, you can see a major rebound of COVID pandemic. So that reminds us the world needs more vaccines, no matter what kind of efficacy level, as long as they achieve certain as US FDA originally required above 50% as predict, and safe, the vaccine will be useful. For that, I will conclude today's Vaccine Congress. Next slide. We hope audience will be interested joining us as a member of ISV to continue our fight against this terrible pandemic. Next slide. We are going to organize, in addition to the vaccine company or product-based Congress, we are also going to organize a special technology-based Congress, such as the mRNA technology. The more information will be available. For those already registered with us today, you will receive from email the future information. For that, I will close today's Congress, and thank you everyone, a special audience from North America. You're staying up to 10:30 at this point. Thank you very much. Hope to see you next time.